Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the evaluation of sedative drugs using Cook's pole climbing apparatus. At the end of this video, we will be able to tell about the effect of sedative drugs or which induces the sedation in animals by using the Cook's pole climbing apparatus. Let us start with the video. At first, let us see the introduction part. The introduction part says the Cook's pole climbing apparatus is commonly used in preclinical pharmacological research to evaluate the sedative effects of drugs. This test is designed to assess the ability of a substance to induce sedation or affect motor coordination and behavior, specifically by observing an animal's ability to climb or interact with a vertical pole. So the introduction part says that this Cook's pole climbing apparatus is used in the preclinical pharmacological research before performing the main clinical part in the preclinical pharmacological research part. And it is used for the evaluation of sedative effects. We are using sedative drugs for evaluating sedative effects of the drugs. And this test is designed to assess the ability of a substance. The ability of a substance means the ability of the sedative drugs to induce the sedation or a motor coordination and behavior for specifically observing an animal's ability to climb or interact with a vertical pole. So, we are observing the animal's ability to climb or interact with a vertical pole. As we can see with the name, the Cook's pole contains a pole and the ability to climb it. Let us learn more about this Cook's pole climbing apparatus. This is the image of the Cook's pole climbing apparatus as we can see here. These are the switches through which we induces the electric current, and the description part says the Cook's pole climbing apparatus is a lab tool or an instrument used in the neurobehavioral and pharmacological research to examine how medications affect animals' conditioned avoidance reaction, usually in mice or rats. So the description part says that this apparatus is used in the neurobehavioral, neurological behavior and pharmacological research. And for what? To examine how medications or the drugs affect the animal's conditioned avoidance reaction. What is conditioned avoidance reaction? Any reaction that is specifically for any condition and for a specific condition, and how does the animal avoids it? And that reaction is known as conditioned avoidance reaction. And we use this apparatus usually for mice or rats. As we can see, these are the rods inside the chamber. This is the chamber of the Cook's pole apparatus. These are the rods through which we induce the electric shock. And this is the lid of the chamber, which is connected through a pole. As we know from the Cook's pole, the pole, which is made up of wood. And the animal, in avoidance for the condition, the animal avoids the electric shock by climbing the wooden pole. And this how it avoids the electric shock. And the pole is made up of wooden because the wood, as we know, that wood is a poor conductor of electricity. That's why we are using wooden pole. Now, the principle. The principle says it operates on the premise that animals may be taught to avoid an unpleasant stimulus such as an electric shock by completing a specified behavior such as climbing a pole. This gadget, okay, is very useful for evaluating possible antipsychotic medications. So the principal part says that this instrument operates or works on the principle that we teach animals to avoid the unpleasant stimulus. What is an unpleasant stimulus? The contact with the electric shock, such as electric shock, we know, and by completing a specified behavior. What specified behavior it completes? It climbs the wooden pole, such as climbing up a pole. So this is the unconditioned behavior or reaction or stimulus that we teach an animal so that whenever electric shock comes, it climbs the pole. And so this gadget is very useful for evaluating possible antipsychotic medications. As we know, antipsychotic medications has a tendency to induce sedation. Now, let us move on forward in our experiment. Now, let us see the equipments required for this experiment. The equipments required, at first, we have the animal. So, we are using rat. The drugs we know, benzodiazepine. The drug we are using is benzodiazepine. And the instrument, as we know, we are using Cook's pole climbing apparatus. Now, let us learn more about what are benzodiazepines. Benzodiazepines are a class of central nervous system depressant or CNS depressant drugs used to treat conditions like anxiety, insomnia, and seizures. So, as we know, benzodiazepines are a CNS depressant. It calms the or lowers the effect in the CNS. Let us see how it works. Benzodiazepines enhances the effect of gamma aminobutyric acid, or we call it GABA in short form, the main inhibitory neurotransmitter in the CNS. So, as we know, that GABA neuron is a neurotransmitter and it is mainly present in the CNS. It is a main inhibitory neurotransmitter in the CNS, central nervous system, and they bind to a specific receptor in the brains, making the nerves less sensitive to stimulation and producing a calming effect. So the so the benzodiazepines affect the gamma neurotransmitter, and they bind to a specific receptor in the brain, and they makes the nerve cells less sensitive to stimulation, or the stimulation of what we call it electric shock, and produces a calming effect. So after administering benzodiazepines, the effect to the stimulation is very less sensitive, and it produces a calming effect for the animals. It does not panics in the situation. So the common uses of benzodiazepines are they are used in anxiety disorders, for insomnia, for seizures, for alcohol withdrawal, for muscle spasms, and for procedural sedation. Procedural sedation are that when before performing any surgical procedure, benzodiazepines are administered to that patient so that they, while performing the surgery, they are calm. Let us move forward in our experiment. Let us see the principle of this experiment. The Cook's pole climbing apparatus is based on the observation that sedative drugs impair motor coordination and behavior. When rat is administered a sedative drug, it typically exhibits reduced motor activity, slower response times. In this test, the animal is expected to climb a vertical pole, and the time taken as well as response to electric shock is also been measured. Serves as a measure of the sedative effect of the drug benzodiazepine. It has a grid floor that acts as a source of shock. Along with that, in the center, there is a wooden pole for climbing. So the principal part says that this apparatus is based on observation of sedative drugs. What is a sedative drug? We are using benzodiazepine. And what effect? It impairs the motor coordination and behavior. It is a CNS depressant, as we know, so it affects on the motor coordination and behavior. So when the rat is administered this sedative drug, it typically exhibits reduced motor activity and slows the response time. It produces a calming effect, so it slows the response time to according to the stimulus. And in this test, the animal is expected to climb a vertical pole. So in this experiment on the Cook's pole climbing apparatus, the animal climbs the vertical pole, and the time taken as well as the response to the electric shock is also been measured. So we record the time taken for the animal to climb to the wooden pole and serves as a measure of the sedative effect of the drug. It has a grid floor, as we have seen in the image of the Cook's pole climbing apparatus. It has grids on the floor, which acts as a source of shock, electric shock. Along with the center, there is a wooden pole for climbing. Let us see the procedural part. The procedure says the rat needs to be trained first in the Cook's pole climbing apparatus. So before performing the actual experiment, we have to train the rats before performing the experiment. So how we have to train it? First, when we will place the animal inside the Cook's pole climbing apparatus, after placing them inside the chamber, the buzzer is pressed. The buzzer is a conditional stimulus. This is the when we will start the buzzer. The buzzer will be alarmed, and after 20 seconds of the shock of the 20 milliampere is given through the grid floor. So after the buzzer, we will induce the electric shock, which will be of 20 milliampere, and we will induce it for 20 seconds through the grid floor. And this is the unconditional stimulus. The buzzer is the conditional stimulus, which which will be assigned before providing them the unconditional stimulus, which is the electric shock. And this cycle is repeated until the rat learn to climb the pole as soon as the buzzer is pressed without getting shocked. So we will repeat this cycle so that the rat learns to climb the pole before providing the shock by listening to the buzzer. So when the rat will listen to the buzzer, it will automatically climb the pole as a sign of the unconditional stimulus, that is the shock. The trained rats are then further chosen for sedative study. Furthermore, the benzodiazepine is administered to observe the sedative effect of the drug. Then the trained drug-treated rats are taken for further study and the activities observed and measured. The conclusion is that by comparing the performance of the treated animals with the control groups, researchers can assess the degree of sedation and evaluate the potential sedative property of new drugs. This test is useful in preclinical drugs screening to understand both the therapeutic and side effects of the sedative substances. So the conclusion part says we are performing this experiment for the control groups. So when we will perform this experiment, we will divide the animals into two groups. One will be of the control group and second will be of the our drug group. In the drug group, we will provide them with the benzodiazepine drug, and in the control group, we will only provide them with the saline solution. And with this experiment, the researchers can assess that the degree of sedation and evaluate the potential sedative property of the benzodiazepine or any new drug. This test is useful for preclinical drug screening and for understanding both the therapeutic and side effects of the sedative substances or sedative drugs. This is the observation table. We will draw this observation table for the experiment. One will be of the control group, which will show, and one will be of the drug treated group. The column says for the response on the buzzer and response on the current. So we will observe the response for the buzzer and the currents. Now let us perform this experiment. And for performing that, we will use our RJPT Informatics Simulatory Laboratory. This is the UI interface of our simulatory laboratory. On the left side of the toolbar, we can see the control cage group, and what on the bottom of that, we can see the Cook's pole climbing apparatus. By clicking this button, we can place them on the laboratory as we've already placed them on the lab's table. On the right side, we can see a syringe which is pre-filled with the drug solution and the saline solution. On the bottom, we can see the instruction part, which will provide us the procedural part according to which we will perform our experiment. On the left side of that, we can see the timer, which will record the time for the animal to climb on the Cook's pole with response to the electric shock. On the right side, we can see the next and skip button. On the top side, we can see the another labeled part which shows the day one of the training for both the groups of the rat. So we have to, for performing this experiment, we have to train the animals. So this portion records the day of that training. On the right side, we can see the observation drop-down menu. By clicking this, we can see the observation table, which we have seen earlier, the control group and the drug group, the response on the buzzer and the response on the current. By clicking this procedural flow button, we can see the flow chart of the step-by-step procedures of this experiment. These are the buttons. These are all the buttons by which we can click them and jump to any step for performing the experiment. By holding this and dragging this, we can see all the steps of this experiment. So now we will start performing this experiment. Then the instruction says take a rat of the control group and put it in the apparatus. So we will take by clicking this take mice button, we will place the control group rat inside the Cook's pole climbing apparatus. We will open the lid. As we can see, this is a wooden pole attached to the lid. And now we will press the buzzer. By clicking this press buzzer button, as we can hear, this is the buzzer. This indicator shows that the buzzer is alarming. By clicking this button, we are alarming. As we can see, the reaction of the animal, that it hurts the buzzing sound and it reacts. Now we will provide the shock. By pushing this push button, we will provide electric shock. As we can see here on the grids, the electric shock is applied and the timer has started for the animal to climb the pole. So after producing the electric shocks, immediately the animal climbs the wooden pole in response on stimulus to the electric shock. We will provide electric shock for 20 seconds, as we can see, according to a timer. And now we will remove the rat of the control group and put it back to the control group cage. Similarly, we will take the rat of the drug group and put it inside the apparatus. For now, we are only training the animals, so we are not providing any drug to the rat. Similarly, we will perform the same process and we will press the buzzer button. We can see the reaction of the animal. We are starting the buzzer, and we are doing it for 10 seconds. Now we'll provide the electric shock. As we can see, in response to the electric shock, the rat climbs the wooden pole. Another reason for using the electric pole is that the rat can easily climb the wooden pole because of the use of its nails. It's non-slippery, and as we can see, the 20 seconds time is up, and we have stopped the electric shock. And now again, we will place it inside the drug group. This was the first day of the training of the animal. Now we will move on to the second day. As we can see here, day 2 of training for both groups of the rats. Similarly, for control group, we will train the rat with the buzzer and the electric shock. We will place it inside the chamber of the instrument, and now we will press the buzzer button. We can see the reaction of the animal with, for the buzzer. The buzzer is placed for 10 seconds, and now after that, we will give the electric shock. As we can see here, the animal climbs the pole in response to the electric shock. By clicking this push button, we induces the shock. And for 20 seconds, we will record the timer. The timer is running as we can see here. And now, after the 20 seconds, we will place the rat back to the control group cage. Similarly, we will do it for the drug group also. We will place it inside the apparatus, Cook's pole climbing apparatus. We will close the lid and we will press the buzzer button. As we can see, the response for the buzzer, the rat hears the buzzer sound and it prepares itself. Now, after giving the buzzer, we will provide the shock, electric shock. The rat climbs the wooden pole in avoidance to the electric shock, and we provide the electric shock for 20 seconds. As we can see here, the timer is running for 20 seconds. And now we will again place it back to its cage, respective cage, which is the drug group cage. This was for the second day training, and now we move on to the day 3 of training for both the groups of the rats. Again, similarly, we will place the control group rat inside the Cook's pole climbing apparatus, and similarly, we will perform the experiment. We will provide the buzzer, and now after beeping the buzzer, we will move on to the next that we will provide the electric shock. As we can see, the rat climbs the wooden pole according to the stimulus for the electric shock. We will provide the electric shock for 20 seconds according to the timer, as we can see here. And after when the timer is completed, we will put the rat back to the rat's cage. This is the control group's rat. So we'll place it inside the control group's rat cage. And now similarly, we will do it for the drug group also. We will take out the drug group's rat. Still, we have not administered any drug to the rat. We are giving them training. Similarly, we will press the buzzer button. For 10 seconds, we can see the reaction for the buzzer to the animal. And now we'll provide the shock. As we can see, in stimulus for the shock, the rat immediately climbs the wooden pole. We will apply the electric shock through the grids. These are the grids, the pole. These are the grids through which we induces the electric current. And now again we will place it back to the drug's cage. Now we have completed the training we have completed the training for both the rats of the control group and the drug group. Now we will check the responses for the trained rat. For that, first we will press the buzzer. As we can see, in response for the buzzer, the rat climbs the wooden pole, which means it knows that when the buzzer starts, after that the electric shock will be induced. So we have successfully induced the stimulus inside the animal. That after listening the buzzer, it immediately climbs the pole before providing them any electric shock. And similarly for the drug group's rat also will perform with the same. We will press the buzzer. As we can see here, in response for the buzzer, the rat immediately climbs the wooden pole, which means the rat knows that after the buzzer stops, the electric shock is provided through the grid. So we have introduced the stimulus inside the rat. And now it's time for the rats to administer the drug. We apply the saline solution for the control group. Take the rat of the control group and apply the saline solution. As we can see, with the help of the syringe, we apply the saline solution through the intraperitoneal route to the rat. And now we apply the benzodiazepine drug to the drug group. We use a different syringe so that there is no chance of any infection for the rats. And now we wait for 20 minutes. This is the stopwatch for 20 minutes. And we wait for 20 minutes for the drug to show its reaction. We can skip this part by clicking the skip button. And now again we will similarly record the response of the drug by performing the experiment. We use the control group rat. We press the buzzer, and in response for the buzzer, the rat immediately climbs to the wooden pole, as he knows that after the buzzer stops, the electric shock is produced from the grids. Now we will remove it from the control group, as we know, because we have introduced saline solution, no drug, only saline solution. And now we test for it for the drug group, in which we have administered the benzodiazepine drug. And now we press the buzzer and see the reaction. As even after listening, as we can see here, even after listening the buzzer, the animal is producing no reaction. It is very calm because the benzodiazepine is a CNS depressant, and after also providing the electric shock, the rat does not climb the wooden pole, which means the drug has shown its effect and it has calmed the animal by providing the sedative effect. The benzodiazepine has produced a sedative effect and made the rat very calm so that even after the electric shock, it does not produce any effect. So now we will remove it from the chamber and place it inside the drug group. And now we have completed our experiment. As we can see here, this is the observation table. So from the control group also, the response on the buzzer was positive and the response on the current was also positive. But in the drug group, after administering the benzodiazepine drug, the response for the buzzer was negative, which means the animal has shown no response. And on the response on the current also, the animal has shown no response, which means our drug has shown its effect, its sedative effect, and it has depressed the central nervous system, CNS, and made the animal calm. So that is also on the buzzer and also on the current, it has shown no response. We can save this response by clicking the Save Observation button. As we can see, this is our observation on observation table. And here we can write our name and our roll number. And when we will click this print button, as we can see here in the format of PDF, we can print and save our response. So now we have completed our experiment. This is observation table, which we have seen earlier. So now it's time for the result. The result says the rats were given benzodiazepine, the sedative drugs, under both stimulus condition, which involve pressing a buzzer and passing a little amount of current, but no reaction was observed, which means when we have administered benzodiazepine sedative drug under both stimulus condition, both stimulus, which means a response on the buzzer and response on the current. These are the both stimulus conditions, which involves the buzzer and passing a little amount of current. But no reaction was observed. As we can see here on the drug treated, no reactions, no response was seen, which means our drug has shown its therapeutic effect of sedation. So now, by performing this experiment, we have learned about the effect of the benzodiazepines on the animals with the help of the Cook's climbing apparatus. So now we have come to the end of this video. I hope that you have learned a lot about this experiment, about the Cook's pole climbing apparatus, and about the effect of the benzodiazepine on the animals. So thank you guys for watching this video. And for more such learning content, subscribe to our YouTube channel, RJPT Informatics. And you can also follow us on our LinkedIn profile, on Instagram, as well as in Facebook. We update and upload our such learning content over these platforms. Thank you.